Breast cancer statistics have shown to vary by ethnic group, with white, non-Hispanic women showing the highest incident rates and African American women showing the highest mortality rates. Traditionally, breast cancer can be classified as either in situ or invasive. In situ breast cancer means there are cancerous cells, but the growth has not spread to breast tissues across the ductal lobule. Invasive breast cancer, which is the more commonly diagnosed cancer, is a type of breast cancer where the cells have spread outside the ductal lobule and moved into surrounding tissues. Traditionally, invasive cancers are what account for the differing stage diagnoses. Breast cancer will statistically affect 1 in 8 women in the United States throughout their lifetime. The diagnosis rate of both types of cancer increased in the 80s, mostly as a result of the increased number of mammograms performed per year. While the screening rate for breast cancer has helped physicians diagnose and treat the disease earlier, additional advancements in the treatment protocol of breast cancer have led to major decreases in the number of breast cancer-related deaths since the 1990s. Research has shown that tumor progression and proliferation is linked to several signaling cascades, most commonly involving estrogen and progesterone-linked receptors. Treatments targeting estrogen receptors have shown high levels of success in decreasing both the proliferation of cancer cells and the size of existing tumors. Unfortunately, a new, more resistant cancer type has also been found, which is not regulated by the estrogen hormone and therefore does not respond to the new treatment therapies. It is believed that about 25% of all breast cancers fall into this class of hard-to-reach breast cancer. Luckily, exciting new research has shown that these cancers contain an overexpression of estrogen-related receptor alpha, which has a unique signal cascade. Researchers are experimenting with drugs designated to target these ER alpha receptors and prevent cancer from spreading. In healthy breast tissue, ER alpha is a steroid hormone receptor that acts as a receptor in a similar fashion to how the estrogen receptor reacts to estrogen. The receptor is 423 amino acids in length and found to bind to multiple response elements. The receptor is classified as a nuclear hormone receptor and part of the NR2 subfamily. Additionally, there was a correlation between the high number of ER alpha receptors and elevated levels of HER2. HER2 is known as the human epidermal growth factor 2 and found to promote growth of cancer cells. ER alpha differs from the estrogen receptor in that there are no known single ligands that modulate its activity, and therefore ER alpha cannot be inhibited in the same way that the current breast cancer drug treatments target and inhibit estrogen receptors. Intense crystallography images have shown that the ER alpha molecule has a binding product cluttered by bulky amino acid side chains. It was found to contain only one nuclear receptor DNA binding domain. This pocket appears to only accept and bind ligands 3 or 4 carbons in length. Additionally, the configuration has shown that the binding region can conform to an active conformation, which induces ligand and cofactors to bind to the ER alpha receptor. Further research has shown that these cofactors can regulate the ER alpha activity by themselves without the presence of the bound ligand. In the pursuit of eliminating breast cancer, current research is being performed into how the hard-to-treat non-estrogen hormone-regulating breast cancer can be eliminated. Steps are being taken to create different synthetic molecules that would bind to ER alpha and work as inverse agonists. In this scenario, the inverse agonists would bind to the ER alpha and prevent production of downstream products, thus preventing cell tumor growth. In theory, the binding of these molecules prevents the ER alpha from having any transcriptional activity. Suppression of the ER alpha receptor has shown to inhibit the growth rates of estrogen receptor negative cancers. A new inverse agonist shown to successfully target this estrogen-related receptor alpha and affect cancer growth is XCT790. This small molecule was found to greatly reduce the concentration of downstream products of the ER alpha signaling cascade. In particular, the XCT790 was found to reduce the protein concentrations of ER alpha as well as to reduce the levels of PGC1. These results were shown using the proteins rHepG2 and HepG2 as markers. In both experiments, there were decreased amounts of the protein as time increased. Additionally, the mitochondrial mass and amount of mitochondrial DNA in cancer cells was found to decrease as concentrations of XCT790 increased. Since ER alpha is involved in energy metabolism, decreasing mitochondrial mass in cancer cells would lead to a decrease in the energy metabolism of these tumors and likely decrease their growth. Moving forward, it will be important to investigate both the upstream and downstream modulators associated with ER alpha to potentially discover new inverse agonists successful in the destruction of hard-to-treat cancer cells.